these all have a lot of acid in them. And these cell walls contain this acid, which helps, just helps them survive. It helps them to have just a strong structure, but it also just gives them this waxy lipid layer. Okay? So this is just another layer of protection that these acid cell wall bacteria have. Okay? So we've gone from outside of the bacteria with, what are the three structures we talked about today? Flagella.
Cilia.
Talked about cilia, but what about for bacteria? Or the more recently? Pili and fimbrium. Okay? So, got to the cell wall, we talked about gram-negative, gram-positive. And then now we're going to go into that cell. So, cell membrane. Our cells are composed of what is called the fluid mosaic model. This means that they can move, they're very fluid, mosaic, they aren't uniform. So we have lipids and we have proteins all in that bilayer. So our membranes, they're bilayers, lipid bilayers. So this is a lipid. You have the head, you have the tails. The heads are hydrophilic, which means they love water, which means they're going to interact great with the outside surrounding, which is usually aqueous, and with the inside surrounding of the cell, which is also usually aqueous. These tails, they're hydrophobic. They don't like water. So water-fearing, water-avoiding fatty acids, they're lipids. So, I mean, you all know lipids and waters don't really mix. Anytime you've had an Italian dressing and you have your oil and vinegar, shake it up, you can get the little tiny bubbles, but soon they separate out into their two layers. So with this, you have just a very nice cohesion layer. You have your hydrophobic on the inside, hydrophilic on the outside. So this way you can kind of protect those hydrophobic in between. Now this makes it so that everything in a way is kind of, the proteins are floating, but also makes a very good membrane because it's semi-permeable. Most things aren't going to be able to go through that because if they're floating free in water, they're not going to want to go through this hydrophobic region because there's a reason they're floating free in water. They look good in water. But we also have these big proteins. The current model is that there's something called rafts, and so these proteins are kind of grouped together in rafts. What they call them. And so you might have a big span of membrane that has nothing, it's just lipids, and then you might have clusters of these neural proteins. But there's still some debate on that one. So it's called a phospholipid bilayer because you have those phosphoheads, you have two layers. More in-depth look of it. So this is a cartoonized version of it. So here you have in blue the heads, which are hydrophilic, hydrophobic. Hydrophilic, okay. The yellow fat inside, hydrophobic. It's a great one to show these integral proteins. So the thing is that you have some proteins which are going to completely span the membrane. Because if you just had lipids, water would never get in. Water can't get through this lipid layer, this fat layer, and we kind of need water. So you have to have some of these different proteins which have multiple functions. Sometimes they give support, structure, but a lot of times they act as channels, letting things come in. We're going to talk about different ways of diffusing or getting things through. So phospholipid bilayer. Peripheral proteins are on the outside versus the integral, meaning inside of it. So if we have our cytoplasm here, outside surface there, does that make sense? Reviewish? Okay, so you can have issues, because if you think about it, we need molecules to move across this membrane. but I just said that water's not going to move across those phospholipid bilayer. But yet, it gets uptake easily. So this is all because of those proteins. And so by having that lipid bilayer, you're able to create a semi-permeable but incredibly selective permeability. Some things get through no problem. Other things have a lot of problems getting through. Yeah. Can you tell me why sodium would have a hard time getting through?
Size.
Yeah.
It's charged.
It's charged. So glucose is just kind of big. So all of these, I mean, they can move across, but it's not the easiest thing. The idea with transport membranes is that we're trying to take something that's outside the cell safely through this phospholipid bilayer inside the cell. Okay? So we have multiple different protein channels that are capable of doing this. A lot of them are very specific. They work for one and one alone substrate, and they will only take that one substrate in. Some of them work with two, and they will take those two, but only those two. So this is how you get, they're called membrane proteins because they're in the membrane. Transport system. So, trying to get stuff into the membrane. You can have facilitated diffusion. So, facilitated or active. Basic difference all really comes down to free. Is it free? Does it not require energy? Or does it require energy? Okay, so there are multiple forms, but the two basic: require energy, not require energy. So facilitated diffusion. So this is something where it needs help. It can't get in on its own, but it wants to go in. So this is where you can use like concentration gradients. So when we start talking about diffusion, if you have a lot of it outside of the cell, not very much of it inside the cell, then it naturally wants to go into that cell. If it has a nice protein channel, which will let it get past that lipid bilayer, then it can go in, move freely, and no energy is required because you're moving with the concentration gradient, so going down that gradient. Now, if you wanted to go the opposite way, that's going to require energy. So that's going to be an active transport. Okay, so if you're trying to kick molecules from low concentration to high concentration, you're going to have to require energy. Okay, does this make sense? Another example would be osmosis. So you can have, osmosis. Osmosis is just a fancy word for, say, diffusion of water, basically. It's just, how does that water move? Facilitated diffusion is what we're looking at. So, you can have some small molecules are actually capable of just passing straight through the phospholipid bilayer. That's pretty small. And just through diffusion. There's more out here than there are in here, they can pass. They're small enough that they can pass, they aren't charged enough so they can pass. Otherwise, you can get facilitated diffusion, where you have an open protein channel, a non-specific protein channel. So you can either be non-specific or specific, and it will just let these, anything the right kind of size, it's going to concentration gradient, fine. You can also have some that are very specific, and they bind to the substrate. And binding to that substrate causes some kind of change, which allows them to be spit out there. So that you can actually get that single protein going through the channel. If you're moving two substances at the same time in the same direction, it's called a symport. If you're moving, sometimes they'll take two substances and move them against each other. It's an antiport. Sometimes science makes sense, sometimes the naming is completely weird. Okay? So, facilitated. Facilitated requires energy, doesn't require energy? What? Not. Okay. active. You have to actively work on this one. So, if we're trying to take something from low concentration into high concentration, it requires energy. It requires a transport protein. A goofy thing that bacteria, I think bacteria are the only ones that seem to be able to do this. It's the transport system. Okay, so active uses ATP, facilitated, no ATP. Transport system. So they can actually alter a molecule as it passes through that membrane so that it can't get out. So for bacteria, they can do this with sugar, so it's a way for them to trap sugar, basically. And so even if it's going against the gradient, they can change it so it can't get back out. I thought it was one of those cool little things that they do, but we don't do it. So yeah, here's an example. You have, so anytime you see the big bright P, it's a phosphate. So usually it comes, it's highly energized phosphate. Usually ATP has that phosphate. And so basically you're taking an energized phosphate and attaching it to your glucose molecule. So now it's not going to be recognized and not be able to be kicked back out. Questions?
So what's this called then?
Okay. Osmosis. So, we can talk briefly osmosis, special case of fusion. Osmosis is really, movement of water across the membrane. And if you have, water is smaller than most other things, so if you have a membrane that is permeable to water but not to the solutes maybe, then what you're going to end up doing is if you start out and you put more solutes, so solute, solvent. What is a solute? Anyone can even give me definitions of these? Do you know what I'm saying when I say solute, solvent? Yeah.
Solute is like cartilage. It's inside solvents that holds the solvent.
Yeah. So, common one that we use a lot is we have the solvent water, and then if you're going to cook some pasta, you throw some salt in there, and that's your solute because it dissolves in that water. Okay. Now, if you add more to one side of this than the other over time, since you can't equal out particles, the water is going to equal out to actually try and balance. Great thing to do if you take a potato, cut some slices of it, throw one slice in pure water and one slice in heavily salted water. What do you think is going to happen to the different slices?
One will shrink, one will expand.
Okay. Since it's a plant, it has cell walls. So, with it's more. Is there a question or comment? The next one. Yeah. So, animal cells are more fun because if you were to throw that much water, so like pure water, around an animal cell, it's going to keep trying to take that water in until it breaks. A plant cell has a nice cell wall, so it'll become really rigid. That slice of potato will become very, very rigid and full. Now, the slice of potato that you put into the super salty water, it's a hypertonic medium. So, okay, so these are important terms: isotonic, hypertonic, hypotonic. And one of the important things to realize with all of these is that they are comparisons. Hypotonic means that the medium outside is hypotonic to that inside cell. So that means, so there are more solutes inside the cell than there are in pure water. So water is going to want to try to go there to dilute those solutes. Okay? And you're going to end up getting a big cell. You can actually make human cells burst because they will try to suck up so much water. Hyper, there are more solutes outside than in, so it's a hypertonic solution. So it's one of the things you have to also make sure what we're talking about. Lots of salt in the water, the solution is hypertonic compared to the cell that was put in there, or the potato slice. And And so in this case, when you put that potato slice in there, it's going to end up becoming floppy. So it's really kind of cool because it kind of shrinks and shrivels. It gets very soft. So very, very rigid slice of potato, very soft slice of potato. Okay? Hyper, hypo. Isotonic. If you found that right perfect spot where you have the same number of solutes inside as outside, you're going to get even flow. The water's going to move in, it's going to move out. Same amounts are going to go both ways. So you're not going to get it to change. So they a lot of times like to show this with red blood cells because, well, they give us such pretty differences. Okay, does that make sense? Iso, hyper, hypo. I personally always thought of hyper, was so excited, just gave everything away. It's my little trick, but however you want to remember them. With plant cells, when you do this, because of that cell wall, they're not going to they'll shrink inside of the cell wall, but the actual cell, not, you know, you're going to see, you can see it just kind of come down, but it still has that rigid cell wall. So the basic structure doesn't move. When it's in a hypotonic, then it's all that entire inner vacuole is going to be pushed up against that cell wall, so it's going to fill that whole space. Okay. Questions? Understand hyper, hypo, iso? So, back to cell structure. Now we're going to look more inside, the internal stuff. So, internally, keep moving in on this bacteria. We have the nuclear area, which is that nucleoid we talked about. That contains the bacterial chromosome. You're going to have some small ribosomes. These things down here, little ribosomes. Bacterial ribosomes are actually smaller than eukaryotic ribosomes. They are, was it 70S versus 80S, and the S just is sedimentation coefficients. So basically you can figure out what is the size of any kind of protein by just spinning them, usually like in a glucose gradient, and seeing where do they fall, you know, what density, how much space do they take up. And so they did that. They found out that prokaryotes are smaller than eukaryotes. They have inclusions, which is where they put, or bacteria just kind of put all their deposits, all their reserves. It's for storing materials. This is, inclusions are one of the problems with using bacteria sometime for making human cell or human proteins, because bacteria goes, I don't know what to do with this, shoves it all into an inclusion body, which we then can't get to, and it's inactive. But for the bacteria, they can put all of their extra food or anything they just want to kind of store out of the way into their inclusions. So you'll get polysaccharide granules, you'll get the metachromatic granules, you know, so they can store their phosphate. You'll get lipid storage. Lipids are great energy sources. Other parts of prokaryotes is the cytosol. So remember, we have our nice pretty cytoplasm. We have what's called cytosol, which is the actual liquid portion of the cytoplasm. So the cytoplasm is that entire space, basically. The cytosol is the actual liquid of it. And then some bacteria can actually create endospores. So when times are so bad that they're like, okay, we gotta do something, they'll actually double their DNA and create this incredibly thick cell coat, and be able to create what's called an endospore. And now endospores are not for mating; they're purely a survival technique. But they've found, they've rehydrated endospores from years and years ago. They're still viable, still able to cause problems if they were pathogenic. So it's one of those cool little tricks of nature to make them survive. Okay. With that. Sizes. Again, just really to reiterate sizes that we're looking at. So, virus, 0.3 micrometers. What's a micrometer again?
-6.
okay, versus bacteria, staphylococcus, versus, you know, a parasitic protozoan, versus the size of a chicken egg. You know, so you can start seeing the difference. I mean, we're talking centimeters, or 47,000 micrometers.
Cray cray.
Eukaryotes. Eukaryotes have a lot more going on. They're more complicated. Okay? So we still have we have a nucleus, and it now has a nuclear envelope. We have a nucleolus, or compact region. It can have some psyllium on the outside of it. Again, we have ribosomes. Okay? Ribosomes are useful for what? Making proteins, translation. Yep. Okay. So we have our nucleus. We have little nuclear pores, so that way we can get this RNA message out of the nucleus so it can get to the ribosome so it can be made into a protein. We have these flat sheets all along the outside of a nucleus called endoplasmic reticulum. There are two types of endoplasmic reticulum. This type that has the ribosome stuck to it is known as the rough ER, or rough endoplasmic reticulum. And then you can have this down here, no ribosomes, smooth endoplasmic reticulum. So this rough endoplasmic reticulum is where a lot of our proteins are made. It's where we get, you know, proteins are put together, they're facilitated, and then they get shipped out to the Golgi. Okay, so the Golgi stacks. You get stacks and stacks and stacks of Golgis. What does the Golgi do?
Transport.
It packages and sends it off.
Yeah, it's thought of as, you know, basically the major packaging source of the cell. It's going to take in things, it's going to modify, it's going to figure out where's everything going, and ships it off, every part of the cell. Some of them it even ships into secretory vesicles and kicks it out of the cell. Okay? We have a cytoplasmic membrane, because you have your cytoplasm, you also have the membrane around the cytoplasm. It's a eukaryotic cell. In this case, it's an animal cell because there's no cell wall. We don't have a cell wall, plants do. Centrioles. These are special to some animal cells, and what they are is they are for the, not mitochondria, but the, I can't remember that name, microtubules. There we go. So the centrioles are important for microtubule assortment. So when the cell is going to go through meiosis, what is meiosis?
Splitting.
So mitosis, cell division, meiosis, going through and creating the haploids. But whenever you're doing cell division, these centrioles move to opposite sides, and the microtubules attach to those, and that's how you actually can separate everything and separate your DNA out to either side of the cell. What else? There's mitochondria. What are mitochondria?
ATP.
Where they make it.
Mitochondria is known as like the powerhouse of the cell. It's where we make the ATP. It's where you make most of your energy. Okay, and they have this. They're double membrane. There are two organelles in eukaryotic, or three. Sorry, but the two major ones that are double membrane. They're special. Do you know which ones those are? So, our nucleus is double membraned, mitochondria is double membraned, and in plants, chloroplasts are double membraned. So, see, nucleus contains all of our genetic information, ATP. If we were talking about a plant, we have chloroplast, which contains chlorophyll, thylakoid stacks, granostacks, everything required for photosynthesis. Lysosomes formed from the Golgi complex. This is what this holds all of the digestive enzymes. You want to be able to, I mean, if a cell dies next to you, that's food. That's nutrients you can use. And so you want to take that in, but you want to break it down into its components, which is what you have lysosomes for, so that you can actually start to break that down and store it. Some eukaryotes have flagella, different structure than prokaryotes, but they do have it. And a lot of them have this cilia, hair-like structure for motility. And I think, let's see, eukaryotes also, they're much larger. So we talked about prokaryotes being what? 1 nanometer basically diameter. These are 10 to 100, micrometers for diameter. Eukaryotic cells have membrane-bound organelles. They include algae, protists, fungi, animals, plants. Another important thing is the cytoskeleton. So if you look at a cell, most of the time, especially in drawings and pictures, it just looks like there's a whole bunch of organelles, and then there's a bunch of just dead space, fluid, which we call cytoplasm. There's actually a whole cytoskeleton, which they kind of tried to draw on the bottom, but it's this whole network of microtubules, actin, tubulin molecules, which gives structure, rigidity. Also, it's how a lot of things move. It's the highway system within the cell. And that's known as the cytoskeleton. And there we will stop for today. So, break, and then we'll come back and talk about lab. So, what time's lab start? 3? 3 o'clock, be back by 3?